Okay, let's start with experiment on photosynthesis. So basically, photosynthesis require carbon dioxide plus water and also light energy in the form of sunlight or other form of light, like normal white light also can, and chlorophyll to give you two products, right? So we can actually experiment on photosynthesis by testing whether carbon dioxide is needed or not, or you test whether water is needed or not, or you test whether light is needed or not, or you test whether chlorophyll is needed. And then one more factor would be you test for whether temperature is suitable or not. So these are the 5 factors that you can play around. Water, we will not play around with water because without water, plant cells will die. All plant cells will die. So we can't really use this as a gauge. So we will only play around with these 4 factors. So before we conduct experiment on this is experiment. Yeah. Where is my blue pen? Okay. So this experiment on photosynthesis. Before we carry out the experiment, we need to do a few steps before and a few steps after. Before the experiment, you have to make sure that the plant is de-starched. What does de-starch mean? De-starch means you remove the starch storage of the plant by keeping the plant in the dark for 24 to 48 hours. You can keep it in a dark cupboard, or you can keep it in a dark room. Okay, the reason for this is so that you will start the experiment with 0 starch. Because at the end of the day, if there is photosynthesis, there will be starch present. If there's no photosynthesis, there will be no starch. Okay? If you never remove the starch storage, you may end up getting a false positive. Because during the experiment, although no photosynthesis occurs, but you test positive for starch. But this starch is probably come from photosynthesis on a few days ago, maybe yesterday or 2 days ago. It's not produced during the experiment. So it is important that you carry out the de-starching steps. Okay? So, Bawan?
Okay.
You understand, right?
Yes.
Yeah, what was the reason for carrying out de-starching again?
Because when you do the iodine test, and if you don't de-starch it and it turns blue black, it might be because there's the starch that's stored inside from photosynthesis a few days ago.
Yeah, it's photosynthesis from a few days ago and not photosynthesis during experiment. So even if you remove carbon dioxide, you will still get a positive result for starch. Okay?
Can, thank you.
After the experiment, you need to remove the chlorophyll because iodine tests, especially, iodine tests require the identification of color. If it is brown color, we know that there is no starch. If it is blue-black, we will assume that we can conclude that you have starch. But the thing here is, the leaf itself has color. The leaf itself is green in color due to the presence of chlorophyll. So you have to remove this green chlorophyll, or else you will notice that the coloration is like brown plus green. Then it looks very dark. You're not sure whether it's brown or blue-black. In other words, if you never remove the chlorophyll pigment, iodine test cannot be observed. So you need to remove the green chlorophyll, and then at the end of the day, you're going to get a piece of leaf that looks something like, a wet tissue paper. Okay?
Okay, thank you.
If it's wet tissue paper, then you have some sort of like white or whitish background. Then when you drop iodine solution, you can see brown or blue-black. Okay?
Okay, thank you.
That is the reason. As for the steps required, you need to carry out a few steps. First, you need to boil the leaf. Why do you need to boil the leaf? You need to kill the leaf. In other words, you need to denature the enzymes. Because if you were to remove the leaf out of the experimental setup, say earlier the leaf was exposed to an area where there's no carbon dioxide. The moment you remove it out of maybe the gas jar or the bell jar, it is exposed to carbon dioxide already. So that means it can start producing starch, can start producing glucose. So in that case, you have to quickly boil the leaf so that you denature the enzyme. Then no further photosynthesis can occur beyond the experimental setup. Okay?
Okay.
So the reason for this is they write it as kill the leaf. Number two, after you boil the leaf, or you also soften it. After boiling the leaf, you will immerse the leaf in warm ethanol. Take note, ethanol is flammable. So you cannot really set up a beaker that contains ethanol and then start heating up like this. You cannot do that because the alcohol will evaporate, and then when it evaporates and reach the fire over here, it will catch fire. Fire will move from here, and then boom, the whole thing will be on flame. Okay? So what you need to do is, you have to set a barrier, a partition. The best partition is, of course, water bath. So say this is your test tube. Test tube contains ethanol. And then at the bottom here, you have water. So this is water. This is ethanol.
The test tube contains ethanol.
Yes, the test tube contains ethanol. And then the beaker contains water. So you are heating the ethanol indirectly.
Wait, then where's the leaf?
Where's the leaf? The leaf will be placed inside here.
Along with the ethanol.
Yes, because ethanol dissolves chlorophyll very well. So in that case, you use ethanol to draw the chlorophyll out of the leaf. Okay?
Okay.
So normally this step will take a few times. Like say for example, you place the leaf in the ethanol. Probably the whole ethanol turn green. Then you take out the leaf, you'll notice that the leaf is less green, but it won't turn whitish ASAP. Okay, you need to repeat this step. You pour out the greenish ethanol, put in a new batch of ethanol, and then put your leaf again. So slowly you draw out more and more due to difference in chlorophyll concentration. So by drawing out more and more and more, and then eventually you'll get a whitish, pale white leaf. Okay, looks like some sort of like tissue paper, wet tissue paper.
Okay, so repeat it a few times.
Sorry?
So repeat it a few times.
Yes, you need to do it a few times until you get all the chlorophyll removed.
Okay.
Then after that, you can do your iodine test. Simple as that. Okay, so the before step and after step is normally not mentioned in the paper. Okay, so normally we are looking only at this, but before and after you have to take note. So once you already done this, you can carry out with the experiment, and then after the experiment you carry out this. Okay, So now we will focus more on the experiment itself.
Then where's the alcohol?
Where's the alcohol?
Yeah.
Ethanol is alcohol.
Oh, okay. Thank
So we start off with carbon dioxide. So for carbon dioxide, you can set up 2 bell jar, both with potted plant. Potted plant of a similar number of leaves. One side, you treat it as a control. It has a beaker of probably water, while this one has a beaker of something that is alkaline. Anything that's alkaline will absorb carbon dioxide because carbon dioxide is an acidic gas. So it could be soda lime. Soda lime is alkaline. It could be potassium hydroxide. It could be sodium hydroxide. Any one of them. Okay?
you. As long as it's an alkaline.
Yes, as long as it's an alkaline. So this will absorb the carbon dioxide. So there will be no carbon dioxide in here. While this one, there will be carbon dioxide. So at the end of the day, when you test for the leaf, after you carry out the experiment for 5 to 6 hours, you start to pluck the leaf out to test for iodine. So you carry out the after experiment procedures, and then you test for presence of starch. You notice that here turn blue black, while this one will remain brown because there's no starch. Simple as that for carbon dioxide. Okay?
Okay.
This one is for presence and absence of carbon dioxide. What if you want to quantify carbon dioxide? Quantify, that means you have different concentration of carbon dioxide. Normally, if you want to quantify something, you use a water plant. This water plant, you put it inside a test tube. The test tube is full of liquid. And what liquid is it immersed in? It is immersed in sodium bicarbonate solution, or you can call it sodium hydrogen carbonate solution. Sodium hydrogen carbonate solution is some sort of like supply of carbon dioxide. So if you have high concentration of carbon dioxide, if you have high concentration of sodium hydrogen carbonate, say 0.01 mole, as opposed to 0.05, as opposed to 0.01 mole per dm cube concentration. This one will have a higher supply of carbon dioxide to the plants as compared to this. It will have 5 times more carbon dioxide supplied to the water plant than this. Okay?
Okay.
So basically what you do is, you carry out, you set up a few sets of this. 1, 2, 3. Say one of them you have 0.01, another one 0.03, another one 0.05 mole per dm cube. So that is how you quantify carbon dioxide. That means this one very little, this one in the middle, this one a lot of carbon dioxide. Then how to find out the rate of photosynthesis?
Is that mole? M-O-L, right?
Yes, mole.
Okay, thank you.
So how you calculate the rate of photosynthesis? You count the amount of oxygen bubbles that are produced. So when photosynthesis occur, the plant will carry out will use carbon dioxide plus water to give you glucose plus oxygen, right? Oxygen is a gas, so it will start to build up in the leaf. So eventually the bubbles of oxygen gets larger and larger. It will start to bubble up. So what you do is you will have to count how many bubbles of oxygen produced. So say for example, this is 0.01 mole, this is 0.03 mole, this is 0.05 mole of sodium hydrogen carbonate. This one probably you get 5 bubbles, or this one you get probably 8 bubbles. This one you get maybe 12 bubbles, something like that. Okay? That means with higher number of bubbles, that means there is higher photosynthesis. In other words, the more carbon dioxide you produce, the more carbon dioxide you supply to the plant, the higher the rate of photosynthesis, the higher number of bubbles produced. Bubbles of oxygen.
Is it possible to use like not use water plants and like use the normal one, but you just like do the same?
If you use the normal plant, number one, you cannot provide it with carbon dioxide. You can use NaHCO3, but it's very difficult to change the concentration of carbon dioxide in the air. Number two, you cannot quantify the bubbles.
Okay.
Because when you test for presence of starch, you can only test whether it is present, is blue-black, or it is absent. You cannot see that, oh, this is very, very blue-black, so it must be a lot of photosynthesis. No, it's either there or no, not there. You cannot quantify the photosynthesis.
Okay, thank you.
Okay. Next one is light. As usual, light, there are two ways. So you either have light or don't have light. So normally you have a potted plant, very easy. This potted plant, you use only the same leaf. This leaf, you take this leaf, you cover up with a black paper. Black paper will absorb the light. So that means this region here, no light, this region have light. So after you carry out the iodine test, you'll notice that this part will be brown, this part will be blue-black, like that. This is presence of light and absence of light. Okay? How do you quantify light? That means you provide more light, provide less light, or maybe medium amount of light. You use lamp. The lamp is placed at different distance. And again, when you quantify something, you use the water plant. Okay, so you can place the lamp within 1 meter, and then or within 2 meter, 3 meter, like that. In that case, you can use the counting of oxygen bubbles to determine the rate of photosynthesis. So you expect the one that is 1 meter to give you more bubbles, probably 15. And then the one is which is of 2 meter probably only have 8. And then the one with 3 meter probably only have 3, something like that. 3 bubbles. Okay? Again?
Okay.
So remember, for carbon dioxide and sunlight, you can quantify it. That means first you carry an experiment where you have carbon dioxide, don't have carbon dioxide. After that, you carry an experiment to get to use low amount of carbon dioxide, medium amount of carbon dioxide, and high amount of carbon dioxide. Light intensity is also similar. Okay, Ken?
What will be put inside the beaker?
What will be put inside the beaker? The one that I showed you just now for carbon dioxide, your plant algae, water plant. And then this is not water. This is sodium hydrogen carbonate.
Why is it still sodium, hydrogen, carbon? Oh wait,
Need
never
to
mind.
provide carbon dioxide for the water plant.
Need to what? Sorry.
Provide carbon dioxide, supply carbon dioxide.
So sodium carbonate.
So for sodium hydrogen carbonate, so for those 1 meter, 2 meter, and 3 meter, they will use the same concentration. You don't change the concentration. The concentration is probably 0.05, 0.05, 0.05. Just that the distance of the lamp is different. Okay?
Okay.
You keep the concentration of the carbon dioxide constant. And then next, we have presence of chlorophyll. Okay, presence of chlorophyll, we can't really quantify it. So we can only use presence and absence of chlorophyll. So for this, we have to search for some plants that are already quite old. Those plants that are already quite old, especially the leaf at the bottom, they will start to have region where it is white in color. That means it doesn't have chlorophyll. The middle one is green. So this type of leaf, we call it variegated. Variegated comes from the word variety. That means it has different variety of color. Okay?
Okay.
So after you allow it to carry out photosynthesis, you will drop it with iodine. You'll notice that the one in the middle will be blue-black. This one will be brown. This one will be brown because of lack of chlorophyll. If there's lack of chlorophyll, then you cannot absorb light. Then you cannot carry out photosynthesis. No glucose is produced. Starch is not produced. Starch is not synthesized. Then there will be absence of starch. Okay?
Okay.
Lastly is temperature. Temperature, we can't really say that there is presence of temperature and absence of temperature, right? Very difficult like that, correct? So what we do for temperature is just quantify it. So again, you set up beaker with your water plant, and then the liquid inside, again, it is sodium hydrogen carbonate. And then probably you put 10 degrees Celsius, 30 degrees Celsius, 50 degrees Celsius. So you maintain the temperature of the sodium hydrogen carbonate. Then you count the number of bubbles produced to determine the rate. You'll notice that probably 30 is the best one. 50 is slightly slower already. Okay, Ken?
Okay, thank you.
Okay, yeah. So basically that's all for this photosynthesis, experiment on photosynthesis.